Molecular dynamics utilizes computer simulations to analyze molecules and proteins at an atomic scale, which helps us understand the world around us and develop useful pharmaceuticals to benefit everyone in the world without stepping a single foot in a laboratory. Recent advances provide free data and software in order to do fast dynamic simulations as well as static discovery. The main software that we'll be using today is called Chimera, and it was developed at the University of California, San Diego in order to do molecular dynamic simulations. However, today we're also going to be using it for just looking at static proteins and understanding the interactions on, between, and within proteins. The software is fast, free, and relatively easy to get started on. I'm sure there are much more advanced techniques that are way over my head at the current level, but getting started really wasn't that difficult. We'll be using several other web-based databases and software, including something called UniProt, which is a database of genes and proteins and provides information on those genes and proteins. Another database that we'll be using is the Protein Data Bank, which is exactly what it sounds, a protein data bank that has information on several different proteins, sort of like UniProt. We'll also be using Muscle, which can align nucleic acid and amino acid segments. We'll use BLAST searches, which can look for nucleic acid or amino acid sequences within the genomes of certain organisms. And finally, we'll also use Swiss model, which allows us to input an amino acid sequence and output a predicted crystal structure of that amino acid sequence based on known amino acid sequences that do have their crystal structure. So for example, we're just going to look at the C3 human protein, which is involved in the complement system, which is a part of the very complicated immune system, which should be conserved between similar organisms. So the organisms that we'll be looking at today include humans, rats, and cows. Specifically, we happen to know that the C3d section of the C3 protein is conserved between humans, rats, and cows. So we can use the aforementioned program Muscle to align the sequences of rat and human C3 protein that we got from UniProt. The region with the most amino acid similarity is the C3d region of the C3 protein. We can then take this C3d section, put it into our BLAST search engine, and search for that amino acid sequence in Bos taurus, which is the genus and species name for cows. Sure enough, one of the sequence matches about 80% similarity, and it's titled C3d in the BLAST databank, so we can be pretty sure that this is the C3d protein that we're looking for. We can go back to the Muscle sequence aligner and align the three sequences now, and that gives us a percent identity matrix. And in this case, it shows us that all of the sequences are about 80% similar to each other. Now we do have the three-dimensional crystal structure of the rat and human C3d proteins, but no one has thought to go and look at the 3D crystal structure of the Bos taurus or cow C3d protein. And so what we can use is probably one of my favorite tools in this video, which is called Swiss model. We can take the C3d amino acid sequence of the Bos taurus and put it into Swiss model, and it will use similar amino acid sequences that have their crystal structures to predict what the crystal structure of the Bos taurus C3d protein would be. Protein folding is a notoriously complex topic and requires intense computational power, so being able to look at similar crystal structures to predict new crystal structures is so cool. It can do it so quickly as well. It only takes about 5 minutes to develop this 300 amino acid crystal structure. So now we have the amino acid sequence and the crystal structure of the rat, human, and cow C3d protein. We see that the sequences are about 80% similar, but what about their three-dimensional structure? We know from things like sickle cell anemia, if something is off by just one amino acid, it can have catastrophic consequences. However, when we put these three crystal structures into Chimera and we use an alignment tool to stack them on top of each other, we can see that the structures are incredibly similar. And this makes sense, of course. The immune systems between rat, humans, and cows are going to be fundamentally very similar to each other. So while we're in Chimera, why don't we take a look at some of the surface features of this C3D human protein? The first feature that we can look at is this area of concavity, which has a group of acidic residues on either side. Another property that we can look at are Coulomic interactions. So for today, we'll look at two different sites and the Coulomic interactions of different groups of amino acids with local amino acids less than 8 angstroms away. 8 angstroms is going to be our top limit for the distance of Coulomic interaction. So we can see at both of these sites, there's several of these Coulomic interactions potentially helping to stabilize the protein. Now, what happens if we bring another protein into this picture? Well, that's the case in the C3D CR2 complex. It's two proteins that have come together. And then what happens to those Coulomic interactions when those two proteins are interacting? Well, in this case, I hypothesize that some of these amino acids are actually helping to stabilize the protein with their Coulomic interactions. You can see the green and the gold amino acids are the C3D protein, and the yellow and blue amino acids are the C3D CR2 complex. And these have been aligned on top of each other, so it's easier to see what sort of changes are going on when the complex is formed. The yellow and the green are the amino acids of interest that we're looking at. And I found specifically that a lysine and glutamine residue from the second group of amino acids that we're looking at actually have new Coulomic interactions that they didn't have previously in the C3D CR2 complex, potentially helping stabilize the entire structure as a whole. The final thing that I want to look at is what would a rat or a cow C3D CR2 complex look like? And so we can do that by superimposing all of the protein crystal structures that we found today onto one model. So here it's a little messy, but we have the rat, the cow, the human, and the human C3D CR2 complex all superimposed on top of each other. The pink is the cow and the green is the rat. And you can see that because their structures are similar in the amino acid residues that we're looking at, we can predict that the stabilizing effect of these residues would likely occur in a rat or a cow C3D CR2 complex. So hopefully I've been able to show today how the power of the free data that's available to anyone, as well as the free, fast, and easy to use software that's now available to anyone, can be used to discover new and novel interactions of proteins all on your own at home without even stepping foot into a lab. Thanks for watching. Bye.